Jen, or Dr. Hellmann, is a brand new minted professor at the University of Dayton in Ohio. And I met Jen when she was still just a PhD student. What happened was that her graduate mentor, Ian Hamilton, and I decided to sort of crush our students together and send them off to Africa to do work on cooperation and cichlid social behavior. And Susan Marsh-Rollo was the kind of auntie or the alloparental caregiver, and she looked after all this big crush of students from McMaster and from the University of Ohio. And that season was hugely successful. There was a 3-month field season. And when the students came back, they had lots and lots of things to analyze and write up. And there was a flurry of about, I think, 14 papers out of that 3-month field season. Of course, it took many years of work later to get those papers out. But Jen was a big driver of a lot of that productivity. And I just wanted to say that she's been incredibly successful. She finished her PhD in about, I think, under 4 years or just over 4 years, which is incredibly quick for a US PhD. The US usually take a bit longer in their PhD times. She continued to have great success. She won an NSF postdoctoral fellowship, which is really hard to get. She also took up a National Institutes of Health and NRSA postdoctoral fellow, and she was a finalist for the Elton Prize, which is given out by the Journal of Animal Ecology. So she's been a huge success, and I kind of understood what a hot commodity she was when potential postdoctoral supervisors kept on calling me up to ask about, what do you think the probability is that Jen will come to my lab as a postdoc? So I knew that we were, she was really, really work with Allison Bell in the Department of Ecology, Evolution, and Behavior at the University of Illinois, Urbana-Champaign, and she's had a very successful interested in the underlying mechanisms for social behavior, and that's the work that we sort of did a little bit together. And in her postdoctoral work, she's been more interested in how stress and parenting influence offspring and transgenerationally how those can shape offspring phenotypes. In her in her current position, she wants to kind of combine all of that together, and I think she's going to be telling us a little bit about that work today. And so we're very happy to have you. Please help me welcome her with a big round of applause. Thank you.
Thanks, Sigal, for that awesome introduction, and thank you so much for having me, and to Susan for inviting me as well. I've been really enjoying the conversations that I've had with people, and I'm really excited to talk today a little bit about the ways in which I've been looking at how fathers influence offspring phenotypes, as particular fathers' experience with different types of stressors. And so when we think about phenotypic variation, is this going to work? There we go. Phenotypic plasticity is this ability of one genotype to produce multiple phenotypes when exposed to different environmental conditions. So you can take two individuals that are genetically identical, put them in two different environments, and they can often appear and behave in markedly different ways. A really good example of this are these Daphnia that develop these helmeted morphs when they're reared in the presence of predators, but don't develop this morph when they're reared in the absence of predators. Similarly, we have things like butterflies and moths that have very different wing patterns when they're reared in wet versus dry environments. And we can also have individuals behave very differently depending on whether or not they find themselves in a dominant or subordinate position in a social group. And so this phenotypic plasticity seems to be a huge source of variation in individual traits. And there's been a lot of interest in this phenotypic variation, in part because it's clear that it alters the ways in which evolution acts within a population. That this plasticity is this rapid response system to allow organisms to cope with environmental change. It may allow organisms to persist in environments that would otherwise be too harsh or changing too rapidly to persist in. We also know that plasticity can often slow evolution via genetic change and alter the ways that genes are operating because shielding genes from natural selection, maybe that plastic response is sufficient in and of itself. And so because of this role of plasticity and evolution and adaptation, there's been a lot of interest in understanding when we see plasticity, how it arises, and is it adaptive? Does it help organisms cope with either natural or human-induced environmental change? And so these are the questions that I'm really interested in, and I'm really interested in how environmental variation drives plasticity in a number of different traits. And so we think about these different types of traits: behavior, physiology, gene expression, morphology. I've been really interested in looking at things on a wide variety of, like Segal talked about, cooperation, but also things like mating behavior and parental care. And I'll talk about today how we see these parental effects altering a variety of different behaviors like anxiety and anti-predator behavior and activity. I'm also really interested in how this is affecting their physiology. How does stress early in life or with parental stress affect stress responses later in life? Hormones, cortisol, or the ways in which individuals are in good versus bad body condition. And then finally, thinking about the ways that plasticity induces changes in the ways that genes are being expressed. So not in the DNA sequences themselves, but in the way that those genes are being expressed, both in early development, but also later in development. And of these different traits, I'm really interested in behavior for a couple of different reasons. So, behavior seems to be very context specific and seems to be a particularly flexible trait. Both physiology and behavior seem to be this inherently flexible trait that can evolve relatively quickly when individuals move into these new environments. That many behaviors tend to be heritable and have really strong fitness consequences. So for instance, the ways that individuals change their behavior when a female comes into their territory, or the ways that individuals change their behavior when they encounter a predator or some sort of stressor seem to have really strong consequences for how well they do in that situation. We also know that behavior is directly linked to the ability to cope with these novel changes that organisms are increasingly encountering in their environment, and that this plasticity can allow for this persistence in new environments and often kind of paves the way and buys organisms enough time to be able to have morphological or physiological changes later down the road. So I've been really interested in understanding behavior in particular. And so generally, as a researcher, I'm really interested in this phenotypic integration of non-genetic information. That we have genes, a genotype that is obviously setting the stage for understanding phenotypic variation, that genes are obviously playing a role, but that we have these other inputs that are coming from the environment. That, for instance, organisms, the environment that they encounter either early in development or currently are affecting their traits. And more recently, we've also started to realize that there's this third source of information, that parental cues or parental experiences can also have really important implications for how organisms behave and respond to their environment. And this is called transgenerational plasticity. So this is ability, or this, when parental experiences alter the phenotypes of future generations. So parents encounter some sort of environmental cue that can affect their state or their information about the environment in ways that then have cascading consequences for offspring. So for example, if we expose these sticklebacks parents, either mothers or fathers, to predators, we see altered anti-predator behavior in offspring. If we in C. elegans, we see that if we expose individuals to pathogens, we have learned avoidance of pathogen behavior for 4 generations after that one-time exposure. And again, this is happening in both moms and dads. So for example, in these mice, this nice study by Diaz and Bressler found that if you train males to associate an odor with a foot shock, such that males start to fear that odor, offspring inherit that same sensitivity to those odors even when offspring themselves have never been trained. So we can get these kind of environmental stressors that then that parents encounter that have effects for offspring. And again, it's important to note that this is not just environmental variation, that's social. For instance, in these squirrels, when mothers are exposed to lots of conspecifics, they have more cortisol that they allocate to their offspring. It causes their offspring to grow faster to cope with this higher competition. And so we have all these different influences or external stressors that are affecting offspring or affecting parents, and these can get integrated into offspring at multiple points in offspring development. And so we can have things affecting, for instance, parental states. Are parents in good or bad body condition? Where do parents decide to lay their eggs or to nest? And then things like epigenetic changes in gametes. We know that eggs have things like maternal RNA. We know that sperm have things like small non-coding RNAs. Those small non-coding RNAs in sperm, for instance, when the egg is fertilized, can repress maternal RNA, and it basically tells the embryo what proteins and what genes to express, and that kind of sets these offspring on different developmental trajectories. So parents can influence their offspring at the time of fertilization. They can also influence them later in development. So, in humans, for instance, there's really big changes in offspring depending on experiences that mothers have during gestation, because it changes the way that they allocate hormones and nutrients to their offspring. And then finally, we can have changes in this post-emergence period where parents can alter the ways in which they're providing care to offspring. And I am particularly really interested in, as I think are many people who are generally skeptical of these parental effects, about how epigenetic cues might be mediated via gametes. So for a long time, as we go throughout our life, we get these epigenetic marks that get put on our DNA that fine-tune the way in which our DNA is being read and expressed. And for a long time, we thought that these epigenetic marks were just completely erased between generations, that offspring essentially started with this blank slate. And increasingly, we are recognizing that that is not true, that some of these epigenetic marks can persist across generations. And this is a really important way that these epigenetic marks and these experiences get incorporated into the germline and can last not just for one generation, but for multiple generations. And so we have all these ways that parents can influence offspring, but I think a really important question is, are they adaptive? What are parents doing here? And there are certainly some instances in which we seem to think that parents are providing offspring with some information about their environment that serves a fitness advantage, that these parental signals have evolved for the purpose of conveying information. So for example, in these Daphnia, not only when individuals are reared with predators do they develop helmets, but when mothers are exposed to predators, they also develop helmets. This is thought to be an adaptive phenotype. There doesn't seem to be any cost, but it serves this really important anti-predator function. Similarly, in these zebra finches, if you expose embryos to these heat calls that parents produce, once embryos hatch, they have higher heat tolerance later in life. So you could rear individuals at at the same temperature, expose one to a playback of a parental heat call, and it turns on some sort of developmental switch that allows them to be more tolerant of heat later in life. This is thought to be some sort of information. Parents are priming offspring about the environment that they're going to encounter. But there's also evidence to suggest that in some cases, and so a really good example of this and why this might be adaptive is if we think about the ways that this might prepare offspring. So for instance, let's say the environment is blue. This could be environment is hot, environment is really risky, whatever. And parents at the time of fertilization provide some sort of cue. Let's say it's some sort of epigenetic mark via eggs or sperm or something that tells offspring that the environment is risky. And this starts them down this developmental trajectory that's starting to prepare them for this future environment. So by the time they encounter this future environment, they have a phenotype that is consistent with this blue environment, right? With this risky environment. On the other hand, if parents don't provide this cue, offspring go about their developmental trajectory, and then finally when they encounter this blue environment, at that point they begin to sample their environment, realize that the environment is blue, it's risky, and then they start to have a phenotypic change that starts to match this environment. But they don't yet have a phenotype that matches this environmental condition. And this could be problematic, especially for organisms that have really high selective pressures early in life, where not matching that environment is really, really costly. This could also be really important for organisms that, for instance, undergo things like ontogenetic niche shifts, where they can't sample their future environment, or it's really risky to sample their environment. So tadpoles can't sample their future environment as adults. Some sort of pathology that then has negative effects on offspring. There are other areas that are a lot more gray, right, where we have some sort of parental cue that is affecting offspring phenotypes, but not necessarily didn't evolve to provide any information about the environment. So for example, if mothers are food restricted, they are smaller, they have smaller body cavities, they have smaller offspring, that has evolved, and then offspring are smaller. We generally think that that might be detrimental, but there are obviously situations in which that might be adaptive. If offspring are born into a low food situation, it might be better to be smaller. Similarly, we know that things like maternal stress induce or make offspring more predisposed to things like ADHD. And so this might be maladaptive if stress is disrupting some sort of developmental pathway, but there might be evidence that this was adaptive in some sort of ancestral environment. So there's a lot of gray area about whether or not these are beneficial, and in a lot of situations what we see is that it might be context dependent, that the ways in which the social or ecological environment in which these are occurring can alter whether or not they're detrimental or potentially beneficial. So for example, if we look at in humans, what we see is that maternal stress is generally bad for offspring. That should come as no surprise. But if you expose mothers to stress and then provide them with lots of social support, we see those detrimental effects going away. That it seems to be context dependent about whether or not it reaches offspring. We can also see, for instance, in a lot of climate change contexts, that sometimes they're detrimental, sometimes they're adaptive. So if you expose parents to a short stressor that's really intense, we see that offspring tend to be kind of crappy. They don't do very well. But if you expose parents to a more gentle stress, so like a subtler change in temperature over a long period of time, that these effects seem to be adaptive in offspring. And so the fitness effects are not always 100% bad or good. But one of the things I'm really interested in is understanding when and how these fitness effects arise. What are these parental effects doing in offspring? And to do this, I've been using three-spined stickleback fish. So these fish are a really famous model in evolution. They're famous for radiating from marine to freshwater environments. They have all of these different body morphs that are associated with environmental conditions, including things like predation risk. So predation is a really salient stressor in these sticklebacks. But it provides a really nice comparative system for looking at different populations, which I'm not going to talk about today. But we also see that there's very consistent individual differences in behavior in response to some of these environmental cues and stressors, and that they rapidly respond to environmental change. They seem to be very plastic. We also find them interesting because they're really easy for lab experiments. They're small, they're easy to house. You can have thousands of them in your lab, and they're pretty hardy, so they're easy to work with. We also know that males and females show really interesting and distinct life history strategies. So males will have these territories, they build nests, they court females. The females lay the eggs in the nest, and then males are the sole provider of parental care. So you can see this male up here, he's fanning his eggs, which is the primary care behavior. He's oxygenating those eggs. And then finally, one of the things that's really interesting is that sticklebacks do show really similar behavior to a lot of these classic neuroscience assays. So things like shoaling, these depth assays, the black-white scototaxis anxiety tests. Sticklebacks show really common responses compared to other types of model organisms like zebrafish. They can be good for these types of questions. One of the things I also really like about sticklebacks is they're a really great system for looking at paternal information and different sources of parental information. So for instance, we can think about the ways that mom and dad influence offspring. If mom is not providing care, mom is primarily influencing offspring in these sticklebacks, because there's no gestation, via changes to eggs. And dads are influencing offspring via changes to sperm, and then dad is also having this additional source of information where he's influencing offspring via behavioral changes to care. And in humans, it's really difficult to disentangle the effects of mom from the effects of dad, because we have these mandatory periods of gestation and lactation where mom is having this really prolonged interaction with offspring. She might be changing that interaction based on dad's phenotype or offspring's phenotype, which is obviously correlated with dad's phenotype. And so it really becomes very difficult to understand what comes from mom and what is coming from dad directly. And so sticklebacks are a really nice system for isolating these different sources of information because they're external fertilizers. So males, you can strip the sperm from the males, you can strip the eggs from the female. Mom and dad never have to meet, so there's no compensatory effect if you don't want it. We can also rear offspring in a cup above an air bubbler, and so offspring don't have to interact with their dad post-fertilization. So we can isolate effects that are coming from sperm from those that are coming from parental care. And generally, I've been really interested in these effects that are coming from sperm. What happens when dads never physically interact with their offspring? The only cue that offspring are getting from their dad is via that sperm and or seminal fluid. And I really like these effects because it's not confounded with early life environment. So when we think about care, how do we know what's coming from dad versus what's just an effect of early life experiences in offspring? And again, it's not, we can kind of completely separate it out from what's coming from moms. So today I'm going to talk a lot about these sperm-mediated paternal effects, specifically kind of touching on, first of all, are these paternal effects specific to fathers? If we expose mom and dad to the exact same stressor, do we see different effects in offspring, or is it just that parental stress generally has the same response? And then I'm going to talk about the ways in which these effects persist through multiple generations. Is it the case that we expose these fathers to a stressor, we see effects in offspring, and then those just go away, or do they persist through multiple generations? And then finally, I want to talk a little bit about the context-dependent nature of these effects, and specifically talking about the ways in which the social environment could both amplify and mitigate these transgenerational effects of stress. So to talk about this first question with the fathers, we took individuals from a California population of sticklebacks. This is a freshwater population. It's a relatively low predation population, but they do have these sculpin predators. It's important to note that predation risk triggers a lot of the same responses, the same physiological responses of things that trigger PTSD in humans. So it is a relatively similar type of stressor in these fish. It is pretty salient. And so we took these fish from these California populations, we brought them into the lab, we allowed them to settle out, and then we isolated males and we had them nest and build nests to show that they were reproductively ready. Once they built a nest, we exposed them to a model predator sculpin 6 times over a 12-day period of time. We chose this kind of short stressor because it's ecologically relevant. They encounter this new predator regime when they move into these shallow waters to breed. We also chose a short stressor because we wanted to, so sticklebacks make all their sperm at the beginning of the breeding season, so we didn't want to interfere with the quality of sperm. We wanted to basically alter, have an effect that was only alterations to already mature sperm. We don't see any differences in fertilization, so sperm are still viable. It's just last minute, these epigenetic changes to sperm. We think it might be these small RNAs that are interacting with these maternal transcripts. So the day after that last exposure, we took the sperm from the male, we used it to fertilize eggs from an unexposed female, and we generated either offspring of control parents or offspring of this predator-exposed father. And so if we see differences among these two treatment groups, this suggests that these sperm is a way in which paternal experiences can influence offspring development. So, but then we wanted to look at, okay, how do these effects compare to what mom is doing? So we then took a third treatment group in which we left males unexposed. We still nested them for that same period of time, but instead we exposed the females to predators. We exposed females in groups. This is because females typically shoal in the wild, so it's natural for them to be found in groups. And did the same procedure where we then did in vitro fertilization. There's no parental care. They're just being bubbled over a cup and to generate offspring of sculpin-exposed mothers. And then finally, we did this double whammy treatment where we exposed both mom and dad to look at the ways in which these effects might be additive. So does having 2 predator-exposed parents have a different effect than having 1? And again, remember, mom and dad never met. There's no compensatory effects going on here. And again, parents never interacted with their offspring post-fertilization. So the only thing mom is giving is eggs. The only thing dad is giving is sperm and or seminal fluid. Then we reared offspring until they were about 4 to 5 months old, and we started assaying them for a variety of traits. One of the things that we looked at is this scototaxis assay. So we can see we have this half white, half black arena. Individuals really don't like being in the white part of the arena. It feels very open and very scary to them. And so we measured how long it took individuals to enter the white section, the amount of time they spent in the white section, how much they switched back and forth as a measure of anxiety behavior. And so what we found is if we look on, look on the y-axis, so we have a PCA of those 3 behaviors. So individuals up here are more anxious. They spent less time in the white area. They took longer to enter that white area. What we see is that offspring of predator-exposed moms show significantly more anxiety-like behavior compared to offspring of control moms. So maternal effects seem to be increasing anxiety in offspring. We don't see those effects in dads. So this is something that's specific to mom. But we do see other types of effects in dads. So if we looked at, one of the things we looked at was survival of these individuals against that same native predators, right? So are these having some sort of fitness benefit? You can see this predator down here. And that's stickleback, who's not very smart and went and approached this predator. So we looked at, does parental experience with predation risk prime offspring, right? Make them more likely to survive a predation event. And so what we saw is when we look at captures of offspring across all these predation trials, so this is on the y-axis here, what we see is that offspring of those predator-exposed fathers are significantly more likely to be captured by the predator relative to offspring of control fathers. So that's not great, right, for them, and that's kind of not what we were expecting and or potentially hoping for. So that's interesting. And I have a couple different theories about why that's happening that I'm happy to talk about later. Is it always bad? I don't know. But interestingly, what we see is that if we look at offspring of predator-exposed moms, we don't see their survival look similar to the control. And really interestingly, if we look at offspring with 2 predator-exposed parents, so both a predator-exposed mom and dad, again, their survival looks similar to the control. So there's something detrimental about having just a predator-exposed dad that's not present when your mom is also predator-exposed and your mom is also stressed. So why are we seeing this, right? Maybe it's possible that the effect of mom is overriding the effect of dad, that both treatment group really resembles offspring of predator-exposed moms. The mom is just kind of this dominant effect that's overriding. And so we wanted to look at that from a gene expression level. So we took brains in these offspring when they were 4.5 months old, and we did pairwise comparisons of gene expression. So looked at differentially expressed genes of offspring of predator-exposed fathers relative to the control, offspring of predator-exposed mothers relative to the control, and offspring with 2 predator-exposed parents relative to the control. And so when we look at just at a gene level, the genes that were differentially expressed, so we can see these white areas are genes that were differentially expressed that were unique to each treatment group. So genes that were differentially expressed in offspring of predator-exposed fathers relative to the control. And then these colored areas are genes that were jointly differentially expressed, that are shared among treatment groups. And we split these for sons and daughters. And what you can see is on a first glance, one thing that's kind of pretty easy to see is that, well, first of all, there's not a lot of overlap in the genes that are differentially expressed. The genes that are differentially expressed in response to maternal treatment are very different than the ones that are differentially expressed in response to paternal treatment and in the both treatment group. And at least at a gene level, a number of gene level, that there seems to be similar number of genes that were differentially expressed across those 3 treatment groups. So at least on the number of genes level, the effects seem to be relatively similar in magnitude. One other thing that's really interesting, I think, is that offspring with 2 predator-exposed parents seem to have a gene expression profile that is distinct from either offspring of a predator-exposed father or a predator-exposed mother. So it's not this idea that mom is just overriding dad. They seem to have very different effects in offspring. But it is important to note that we do have some shared genes in this middle here. And when we look at those shared genes, what we see is that they are differentially expressed in the same direction. So in red, we have genes that are upregulated. In blue, we have genes that are downregulated. And what we can see is when genes are downregulated in response to paternal treatment, they're also downregulated in response to maternal and both parents being exposed. So when genes are differentially expressed and shared, there's this core set of genes that seem to be shared among these treatment groups. And I want to note that we did separate this out for sons and daughters because we see really strong sex-specific effects that I'm going to talk about in a second. Okay, so hopefully I've shown you here that these paternal effects do seem to be really unique to fathers. Dads are not doing the same thing as moms, and that there seems to be this interaction between maternal and paternal cues that I think is really important. Okay, so what about these effects persisting through multiple generations? Is it the case that these just go away, or do we see some sort of lingering effects? And I think that this is a really important question because there's a lot of talk about when would it be adaptive to maintain phenotypic change across generations. So if the F0 generation, if those dads were exposed to a predator and then their offspring never saw a predator and then they had grandsons, why should the grandsons listen to the experiences of their grandfathers and prioritize that over their experiences of their fathers who never saw a predator? Why continue to maintain this anti-predator phenotype in the absence of reinforcement? I think conversely, this question is really interesting when we think about deleterious experiences, so things like toxin exposure. If we expose one generation to toxins, for how long do we continue to see persistent effects of that toxin exposure? If we clean something up, how long does that negative effect persist? Multi-generational persistence across multiple studies, that we do see a lot of evidence that phenotypic changes generally do persist across multiple generations. And in some cases, these effects persist to all individuals. This is true here. We have some studies in C. elegans, for instance, and rice and mouse. And a lot of theory predicts that it should kind of be this all or nothing phenomenon. Either it persists to the second generation or it doesn't, and that's it. But the reality is that we actually don't see that a lot happening, that in a lot of cases, actually these phenotypic changes persist selectively to the second generation in a subset of individuals. So for an example, in humans, it seems to be sex and lineage specific. So in this Dutch hunger famine, what we saw is that when grandparents were exposed to famine, that it affected, basically, grandsons are affected by the diet of their paternal grandfather. Granddaughters are affected by the diet of their paternal grandmother. So it's sex and lineage specific. This is echoed also in a number of studies looking at mice. But all of these studies have been done looking at these mammals, right? We really don't see a lot of studies looking at sex and lineage effects in organisms that don't have these mandatory periods of gestation, right? In gestation, we know there's sex-specific placental function that could be generating these specific differences across lineages or between sexes. And so we were really interested in understanding, do we see these sex effects, these lineage effects, when we don't have gestation, when mom is not interacting in the same way with their offspring? And so to look at this, we took the same sticklebacks from California and again had that same paradigm where we had this short stressor that we exposed this F0 male to, right? Some males were stressed, some were not. We used in vitro fertilization to generate those F1s. We reared the F1s in the absence of predation risk and then bred the F1s to generate a second generation. So for instance, we bred sons of control fathers and daughters of control fathers to get F2s with control grandfathers. We bred daughters of control fathers with sons of predator-exposed fathers to get F2s with a predator-exposed paternal grandfather, so just via the paternal lineage. We bred daughters of predator-exposed fathers to sons of control fathers to get F2s with just the maternal grandfather exposed. And then we bred sons and daughters of predator-exposed fathers to get F2s with two predator-exposed grandfathers. To again, look at both these lineage effects. We sexed all the offspring so we could see what F2s are male, what F2s are female, and do the effects influence those sons and grandsons and granddaughters differently. And so we assayed them when they were about 5 months old. So the sticklebacks mature at like a year. So they're still juveniles at this point. And when we look at a bunch of different traits, we see really strong evidence for these sex and lineage specific effects. So here is the change in activity and exploration when individuals undergo a simulated predator attack. So individuals high on this axis are really responsive to that simulated predator attack. Individuals that are low on this axis kind of just ignore it. They just go about their business. And what we see is that when we look at grandsons, there's not really any effect of paternal treatment. However, or paternal lineage treatment of grandfathers. However, when we look at daughters, granddaughters that are in red, what we see is that granddaughters that have a paternal grandfather exposed to predation risk are significantly less responsive to the simulated predator attack compared to the control. So female F2s are being affected by the experiences of their paternal grandfather. And this is also true not just for this trait, but also for something like mass. So again, if we look at mass of these F2s, what we can see is for grandsons, doesn't really seem to be an effect of grandfather experience. But we look at these granddaughters, what we see is, again, this really strong effect of paternal and maternal experience, grandfather's experiences. So these females are being affected via the paternal lineage. This seems, a bit out of place, right? Maybe not necessarily what we would have predicted, but it is actually really consistent with what we see in a lot of the mammalian literature. That consistently what we see is that females are being affected, and it's only transmitted via the paternal lineage. So males are silent carriers of these inherited phenotypes. We see this in response in both humans and in mice, and we see this in response to a variety of different stressors. So I think it's really interesting that we see these same effects in sticklebacks, despite the fact that in all these mammalian studies, the Q originated in moms. But this matching, is mom telling you that it's dangerous and dad's telling you it's dangerous, you're getting the same cues, or is there different cues that might increase uncertainty about the environment that you're going to encounter. Okay, so collectively, hopefully, these effects suggest that we see these effects persisting through the F2 generation in both lineage-specific and sex-specific ways, and that there's these important interactions between maternal and paternal lineages. And so finally, I want to kind of end talking about the ways in which these effects might be context-specific. Okay, so we, and really trying to understand how does the social context influence the evolution of transgenerational effects. Oftentimes when we do a lot of these studies, individuals are isolated to control for experiences. They're not in a social group. But the reality is that most individuals spend the majority of their life in a social group. And this social group is a really important mediator of stress and the ways that individuals interact with their environments. So take these birds, for instance. We know, for instance, that the ones that nest on the edge are much more vulnerable to predators. They're going to be more potentially stressed compared to individuals that are in the center that are more buffered by their social experience. And so we have a lot of evidence for social effects within a generation. And so we think about how sociality can mediate stress responses. If we have this one individual in a group that experiences some sort of stressor, it's possible we have no social effects. The other individuals just remain unstressed. That individual that experienced the stress remains stressed. There's no transmission effects. But we could also see something happening where we have this stress buffering. That individuals experience some sort of stressor, they go back to their group, the rest of their group is unstressed, and so that helps bring that other individual back down to baseline. Alternatively, we could have some sort of stress transmission going on where that individual is stressed, he goes back to this group, the other group members are like, oh, there's something stressful here, and those other group members become stressed. And so we have evidence for both of these things happening within a generation. So for example, there's a lot of evidence for stress buffering in both humans and non-human animals. So humans, we see that social interactions can bring glucocorticoids down very quickly after parents experience some sort of stressor, or individuals experience some sort of stressor. Similarly, in these baboons, or so these monkeys and birds, we see things like allogrooming and allopreening that are these affiliative behaviors that reduce cortisol. So it provides some sort of buffering effect via social interactions. We also know that there can be stress transmission. So a really nice example of this is a study in mice where they took these 3 individuals that all lived together. They took this green mouse out and they exposed him to a foot shock, which is obviously very stressful. And then they put the green mouse back in with his litter mate, this blue mouse. They allowed them to interact for 30 minutes. Then they took the blue mouse and put the blue mouse with the orange mouse. And what you can see is that all 3 individuals show these really distinct changes in brain activity that's associated with stress. So this blue and this orange mouse never experience the foot shock, but yet become stressed because of this transmission effect that we think is being mediated via sniffing and olfactory cues. So what I would argue is an underexplored possibility is not just that these effects are having, these social effects are affecting within a generation, but they also might affect the ways in which stress is mediated across generations. That maybe this buffering effect could mitigate TGP. If moms become stressed, but then the stress goes away very quickly because of those social interactions, maybe offspring are not adversely affected. Similarly, if we have dads becoming stressed, or individuals becoming stressed, they stress and infect other individuals with that stress, then we can have this proliferation of stress across different individuals. I think that this is really important for thinking about neighborhood effects and, for instance, humans, and the ways in which positive and negative influences are transmitted across individuals. And then I think, thinking about the interaction between external stress and internal response. That you could have 2 individuals that experience the same external stressor, but could internally respond in very different ways that mediate whether or not these have downstream effects. And I think these social stress transmission effects are really important because it suggests that these transgenerational effects are reflecting not just the experience of one individual, but of multiple individuals in a social group. That you don't have to experience stress directly for your offspring to be affected. And so I want to go through both of these examples and data that we found through both of them. And so going back to this original diagram that we had where parents are experiencing some sort of stressor directly that then affects offspring, this could be social or environmental. We increasingly have evidence that that we have this connection between conspecifics giving some sort of information about the environment. So this can happen in the context of these mice where one mouse is stressed, it comes back, and the other mice pick up on this idea that the environment might be stressful. This can happen in fish, for instance, with anti-predator behavior, where if an observer or demonstrator is exposed to predation, they have anti-predator behavior. This triggers anti-predator behavior in an observer fish. So individuals are picking up on cues in their environment and stress in their environment. And they're doing this, so an underexplored possibility is not just that they're doing this within a generation, but again, this might be transmitted to offspring. That if non-parents experience a stressor that induces some sort of change in olfactory or visual cues, the parents then pick up on this, it changes their state or condition or information about the environment in a way that then affects offspring phenotypes. I think sticklebacks are a really great system for looking at these questions. As I hope I've kind of shown you today, we have really strong evidence that we have this vertical transmission, that stress in the fathers then affects offspring. We also know that their social system is really well set up, that they're paying attention to other conspecifics, that individuals can detect predator-exposed conspecifics, say males will court predator-exposed, females less. And that these individuals are paying attention to each other, that males are paying attention to females because they're courting them, that males are paying attention to other males because they're interacting across these territory boundaries. So there's definitely opportunities for this information, these cues to be transmitted between individuals. So to look at this, we've simultaneously manipulated paternal exposure to predation risk and exposure to conspecific cues. So we had males in these tanks, and some of these males were predator naive. They had a neighbor who was predator naive, so there's no predator cues going on here. They're just interacting with each other. We had these directly exposed males where the males were exposed to a model predator, but their neighbor was predator naive. They received olfactory cues from unstressed males. So they're getting visual and olfactory cues of unstressed males. We then have individuals who remained predator naive. They never saw a predator, but they exposed their neighbor to a predator. Importantly, there was a barrier between the male. The father never saw the predator himself. They saw his behavior in the aftermath of that predation event, and then that father received olfactory cues from stressed males. And then finally, we had this double whammy where males were stressed directly and their neighbor was stressed to look at the combined effects. And really what we were looking for was, do we see differences between these two males? If so, this suggests that there might be some sort of stress transmission or information transmission. The males are altering their transgenerational effects or what they're putting in sperm based on their neighbor's experience. And so we assayed larvae when they were 3 to 4 weeks old. And what we saw is that when we look at these larvae assay, we look at freezing time after a simulated predator attack. What we see is that offspring with these neighbors who were predator exposed, so dads never saw a predator themselves in this indirect group, just the neighbor, then we still see this really strong reduction in freezing behavior in the offspring. So this suggests that dad does not need to be exposed to a predator to induce changes in offspring phenotypes that's sufficient for his other individuals in the social group to be exposed to predators to induce these transgenerational effects. We also see similar changes in things like and so it is, again, what you'll notice is one of the things that decreases is this it's a decreased anti-predator response rather than an increased anti-predator response. And again, this is consistent with this risk allocation hypothesis. If the environment is risky, just ignore it and keep going on and be high risk, high reward. And again, is also consistent with some of the other effects that we've seen in this F2 study that I just talked about. What we see is that F2 males whose grandfathers were exposed show less time freezing, so they are resistant they have this reduction in anti-predator behavior. And I think I'm going to skip ahead. I want to kind of make sure we have enough time here. We see this similar pattern with indirect exposure for the larvae. for mass as well. So again, this effect of the neighbor being exposed. And so again, we have this change in anti-predator behavior or cortisol. We think both olfactory and visual cues are important. They're changing potentially some sort of cue or state, inducing some sort of parental stress or providing fathers with some sort of information about the environment that in our case seems to be passed down via sperm and or seminal fluid. And again, suggesting that offspring phenotypes seem to be influenced by two non-genetic mechanisms happening simultaneously. That we have maybe this social learning or there's stress transmission within a generation from male to male, that then is passed vertically from males to their offspring. So multiple non-genetic mechanisms are important here. So we have this stress transmission being really important. What about the stress buffering effect? I'm going to go over this really quickly. We know that social isolation is really important, that these social interactions can help attenuate the stress response, bring individuals back to baseline. The individuals who are socially isolated stay stressed for longer. And so we wanted to do this in the FESH and look at how the social isolation altered the transgenerational effects that are mitigated or induced by predators. So we manipulated both social isolation and predation experience. So the design looked a little bit different this time. We had this model, we had this live trout predator in a nearby tank for 2 hours a day for 10 days. We pulled this barrier so sticklebacks could see this predator. They received these trout cues. The trout were being fed the stickleback fry during this exposure. So they got these visual and these olfactory cues of these predators. If they were controlled, they got an empty tank and they just got plain water. And for some of these males, they saw the predator when they could interact with this male neighbor. There was ability to get visual and olfactory cues passing between them. But for some males, they saw this predator when there was barrier present, so they couldn't see this other male in the tank. There was no transmission of olfactory cues either. And then we, again, did in vitro fertilization, took our sperm, looked at offspring development or offspring traits. One of the things that we looked at was survival against predators, this time in a much bigger arena that's potentially a little bit more realistic. This is work done by my grad student, Michaela. And when we look at offspring survivalship against predators, what we see is offspring survival on the y-axis, so individuals survived for a long time or got eaten very quickly. What we see is social isolation seems to have a pretty big effect, that individuals who were exposed to predation risk while they were isolated got eaten much quicker than offspring than the control with the neighbor. And it's really this effect of social isolation that seems to be important. That even the control with the social isolation seems to get eaten much faster by the predator. So there's some sort of detrimental effect of being isolated that is potentially, carrying on these effects. And maybe not the predator stress itself that's stressful, but it's the isolation. And I should note that all the studies we had done up to this point, the males were isolated. So, do with that what you will. Okay, so hopefully I've shown you that the social environment can both mitigate and induce transgenerational effects. It's really important to account for this social environment. And so, overall, we see that maternal effects mediated via sperm seem to be different than what mom is doing, and that there's these really important interactions between mom and dad that it's important to account for. We can't understand dad without understanding mom, and vice versa. We also see that there's these really important lineage effects, that these effects persist through at least 2 generations, even when the affluent generation never encountered a predator. And again, we're seeing these really important interactions between maternal and paternal lineages. And then finally, the social environment seems to be really important, that so other individuals can either help buffer parental stress in ways that kind of stops the transmission or mitigates the transmission to offspring, or other individuals can induce parental stress in ways that creates transmission of these stress effects. That maybe means that stress effects these transgenerational effects are reflecting the experiences of entire groups, not just of an individual. And so with this, I'd like to thank all the individuals that helped out with this research and all the really valuable undergrads and my funding sources, and I take any questions.
for some questions, and I'm also inviting people after the talk to come upstairs and continue to ask Jen questions in the social. So let's start off with Katrina.
Thank you for the nice talk, really interesting data. I have a question regarding the very last set of experiments you've shown regarding social isolation and buffering. I'm just wondering the heightened risk of predation and socially isolated fish. How much of that do you think could be potentially due to just more like heightened drive for social investigation or interest? Just to see another moving animal in the arena might drive the socially isolated fish to approach.
Yeah, so that's a good question. So I don't know how much males are changing their behavior other than their behavior when they're isolated. I will say that when we put them in the arena, they were with another individual. So they weren't alone in the arena, so we had 2 individuals in the arena at a time. So we just recorded time until the first one got eaten in order to basically have that opportunity for social interactions. And so we still see these detrimental effects even when other conspecifics are present. Now it's possible that social isolation in the father could change those social interactions in a way that makes them like less useful or maybe less able to kind of use each other to avoid predation. I'm not sure if that answers your question.
Thank you, and what a great model because you can really separate out the gestational effects. I'm just curious as to whether you can speak to whether some of the predatory experiences, does it matter when the parents experience it, or is there a bigger impact if it's closer to the time that they're creating sperm and eggs?
Yeah, I mean, it's a really good question. I mean, the short answer is we don't know. We've only ever done immediately prior to fertilization, again, in order to try to minimize the amount of potential damage to the sperm that we've been doing. I mean, this fall or this past year, we did like a really long-term exposure. There were issues that I'm not sure we're going to be able to suss out what's going on, but long-term exposure seemed to really have very strong influences on things like size, and so then, would probably have effects on offspring. And we know that we see these effects even if we only expose parents for 3 days, so you can have a really short stressor and still observe the effects, but whether or not we have like a lag time effect, like if you expose parents and then waited a month and then took off, we don't know. And there's definitely effects in some species for reversal, not necessarily attenuation, but like if you, for instance, in that mouse study where they paired the foot shock with the odor, if they take the sperm right away, they see effects in offspring, but if they reverse the effects, they keep giving the they keep exposing the males to the odor but no foot shock, so they become used to the odor and it's not scary anymore.
Right.
And then they take their sperm, then there's no effect in offspring.
Right.
So we do see some evidence that might happen.
I guess also because you showed that there are effects of isolation and social buffering, the longer the leg, those other things also impact whether it was a stressor or not.
Yes.
So it makes it difficult.
Yeah.
More of like a personal interest question. So you mentioned kind of in the introduction that you can evaluate these transgenerational effects in response to all sorts of things. Like you very thoroughly tested it with stress here, but you mentioned anthropogenic effects, maybe like heavy metal contamination in aquatic systems, or relevant to Hamilton's sewage contamination. We just found out that there were like decades of waste being dumped into Coots Paradise. If you had to pick a new direction of stressor to start exploring these transgenerational plasticity effects, and where would you go?
Ooh, that's a good question. I'm really interested in kind of separating out like what is information versus what is state, and I think like the predator that we've been using is kind of a mix of both, and we don't really know what is going on. Is it state change-based changes? It's just like stress is inducing some sort of physiological change, or do individuals have some sort of information about the environment? And so I think moving forward, I'm really interested in kind of picking a stressor that's either purely information or maybe purely like very clearly kind of stress-based to kind of suss out those effects. So something like what I talked about yesterday with a juvenile predator that's not scary, so the dad doesn't eat the dad, but is relevant to the offspring. I think that's a good example of information. I think those heat calls in zebra finches, I can't do that in fish. That's a really good example of information. Toxins, I think, are pretty uniformly stressful. There is a little bit of evidence that parental toxin exposure can increase toxin resistance in offspring. What we've been doing, we're starting to do some glyphosate exposures, so that's an active ingredient in Roundup, and we've been doing some heat experiments as well. So, I don't know, we'll see if any of those things become interesting.
Thanks.
Are there any other questions? Go ahead, Zoe.
I was wondering, in the first study where the mothers were exposed but in groups and you didn't see an effect of maternal, only paternal,
Yeah.
do you think you would still see that if the mothers were also isolated?
Yeah, and so I actually really thought about that, but obviously we know that social isolation is really important. And so when we were designing the experiment, we thought about that. Do we isolate mothers so the experience is really parallel to fathers, and the social environment is really parallel to fathers, or do we keep it ecologically relevant, right, and expose them the way that they would be exposed in the wild? And my gut is to say it probably would have been different if the mothers were exposed. The ways in which it would be different, I don't know. I think there's, and I think the fact that we use in vitro fertilization probably also changed things. I think there's probably really important interactions going on in mate choice and parental care that are kind of mediating this interaction between mom and dad that we haven't yet captured. So it's a great question, and we don't know yet.
Any more questions? Okay, well, I guess I'll just say thank you for that tour de force. It was absolutely wonderful. A little terrifying when you think about the fact that Jen showed her fish stressors for like 6 times over 12 days, and you think about how much stress we have every day in our lives and what that's doing. But thank you very much, Jen. That was really, really wonderful. Let's all give her a And don't forget, there's going to be lovely food upstairs on the 2nd floor, and you're all invited to come and have a little bit of treat and ask Jen about some of her studies and future directions and so on and so forth. I hope to see everybody